Okay, so first, this is the whole microbiological lab, that is the whole work which are done in a microbiology lab. So first is the media preparation. So first step is the media preparation, which is done in microbiology lab. So any microbiological analysis. So first is the ready-made media which we can get in market. So we will take the particular ready-made media for growing of the particular microorganisms, and then we will measure or analyze or weighing that media in a weighing machine. And after that, after weighing the particular quantity of the ready-made media, then we will pour it in a measuring cylinder or conical flask or beaker. And after that, in the beaker where we take the ready-made media, we will add the particular amount of distilled water or double distilled water or deionized water. We will mix this deionized water or distilled water to our particular media, okay, or particular amount of media, and then we will mix and we will prepare the media. Okay, so there this is ready for our our media is prepared for next step. So the next step is media sterilization. So how we will done the media sterilization? So media sterilization is done by autoclave. So through this autoclave we will sterilize our media, so no microorganisms are contaminated or no microorganisms are present in our media. So media is totally sterilized for testing or for next step. So after media sterilization, we will go to the inoculation steps. So here in inoculation, so for inoculation we use equipment or instrument, that is laminar airflow, which provide us the sterile environment where we perform our testing or analysis of microbiological organisms. Okay, so after inoculation, so where we inoculate our culture or microorganisms, so we inoculate our culture or microorganisms into our media which we have sterilized in this step. Okay, so after sterilization we inoculate our microorganisms in the inoculation step, and then we incubate our inoculated media into the incubator. So in incubator, particular temperature and particular time is maintained in the incubator for incubation of the microorganisms. Suppose for some bacteria 37 degrees Celsius for 24 hours is required, or some different types of microorganisms for their growth they required 42 degrees Celsius or 25 degrees Celsius temperature for 24 hour or 48 hour or 72 hours. So different types of microorganisms require different types of incubation condition for their particular growth. Okay, so after incubation or after growth, then we will see the observation or the microorganisms whether they grow on our particular media or not. So here we will, in the petri plate we will see the colonies appeared after incubation. So we will count these colonies in colony counter, and after counting colony counter or after using colony counter we can observe the result. And if we want to measure or if we want to see the morphology of the microorganisms, then we will use microscope and we can capture the data in our computer. Okay, so this is the whole process. So first step is the media preparation, next step is the media sterilization, and third step is inoculation, fourth step is incubation, and the fifth step is observation. So this is the whole step of a microbiological lab for microbiological analysis. Okay.